Welcome back, students. This is Module 4.3.5, Patterns of Evolution. And although I'm titling it Patterns of Evolution, there are several concepts that we're going to discuss before we get to Patterns of Evolution. The first involves this quotation by Stephen Jay Gould. He was an eminent evolutionary biologist at Harvard University. He died of a rare form of cancer several years ago. And he said, "Ideal design is a lousy argument for evolution." And we can explain the sense of this statement. This is because natural selection acts upon existing structures and it produces odd arrangements and funny solutions. So natural selection doesn't go back to the drawing board and design things from scratch, but it uses these existing structures. And so rather than expecting ideal design, we should actually expect non-ideal design. So non-ideal design or imperfect structures should be expected, and this type of design provides evidence in favor of evolution, as structures are often repurposed. Some examples of non-ideal design. For example, the fact that the majority of human DNA doesn't code for proteins, and much of it is what we would call junk DNA, at least half of it. Certainly there are sequences of DNA that don't code that are regulatory, such as promoter regions and other things, but that still doesn't account for the large proportion that doesn't seem to do anything. Another example of non-ideal design would be the panda's thumb, and we'll talk about that in a moment. And also the human eye. So let's look first at the panda's thumb, and then we'll turn our attention to the human eye. Pandas are bears. They're members of the bear family, and they're in the order Carnivora. To be a carnivore doesn't mean you have to eat meat. It's actually a genetic lineage. So they're related to the lineage of meat eaters, but pandas, of course, are really pretty hardcore herbivores. They will opportunistically eat baby animals, but they don't chase down other animals and eat them as many carnivores do. In fact, a panda may eat 30 pounds of bamboo each day, and they sit back on their haunches, as pictured here in the upper left, and they strip the leaves off. If you've ever felt bamboo, it's very silicaceous. So silica is actually what sand is, and so it has like this, it's very low in nutritional value, so they compensate by eating large amounts of it. And as it turns out that most vertebrate animals, including members of Carnivora, are pentadactyl. That means they have 5 digits on their limbs. And if we see like a grizzly bear here on the left, and we can see the digits numbered 1 through 5, and there they are. But if we look at a panda, we see 1 through 5, but then there's another little thing over here, and this is called the radial sesamoid. It is a small wrist bone, which in pandas has evolved to become this large flap-like contraption that's used to strip leaves off of bamboo leaves. Off bamboo shoots, excuse me. And so this actually shows how evolution can behave, how it can take something that is already there, a wrist bone, and repurpose it. And of course, if we're going to design a thumb for a panda, we might, as humans, take finger or phalange number 1 and make it opposable, kind of like ours is. But that's not what happened in the case of pandas. And we can suppose that there were a series of steps in which a little bump on the wrist got bigger and bigger, and those with larger bumps could strip leaves a little more efficiently, and so they survived and reproduced at a higher rate. They had more energy to do so. Until today, when we look at a panda's foot, we can actually see there's 1, 2, 3, 4, 5. Those are its digits, and this is the panda's thumb that's opposable and used to strip leaves off of bamboo shoots. So pretty remarkable example. Let's look at a second example. This example is closer to home. This is the human eye. And my figure doesn't look like it's working very well, but nevertheless, we can see in cross-section in the upper left here, the human eye, and we can see the lens, and light would shine through. And on the back of the eye, there's a light-sensitive layer called the retina. It's made out of nervous tissue, and that's kind of what's being shown here. These little cells represent nerve cells, And what it shows is the light is coming from the right, although again the animation is kind of screwy, but just bear with us. Right there is a view that shows there's the body of the nerve cells, but the wiring of the nerve cells doesn't go behind the light sensors, it goes in front of them. And so it's kind of a weird way to design a light sensor or an eye. And when all of these axons of these nerve cells coalesce and come together, they form something called the optic nerve, which is now inside the eyeball, and it has to go to the brain, which is behind there. So it passes through the retina, the light-sensitive layer, and as it passes through where it is, there can be no light receptors, and this leaves the human eye with a blind spot. And so the blind spot results from non-ideal design. This is not the way any engineer would design a light sensor. If we want to see a more, quote, ideally designed, in quote, eye, we can look at the eyes of an octopus. And this is showing, they have the same type of eye. It's kind of the design of a pinhole camera, small opening where light comes in, and then we can see an image form on the back. And in this case, this is showing a squid eye, and it shows that the nerve fibers or the axons of the nerve cells come behind. This is the way an engineer would design, and the light falls on the light-sensitive area right here. And the result is cephalopods, the group to which octopuses and squids belong, do not have a blind spot. And this actually shows a direct contrast. So if you had trouble with my animation, it wasn't performing properly. We can see here that in this case the light is coming in the opposite direction from the right, and we can see in the human eye that all the nerve fibers are in front of it, and as a consequence they pass through the back of the retina and create a blind spot. Now you can actually see your blind spot on your computer screen. This doesn't work when I project this in the classroom, but it works pretty well here. So follow the instructions down here. It says to see your blind spot, close your left eye and stare over here at the plus, and move slightly closer or further from the screen, and you'll get to a spot where there's the O just disappears, and right now I can't see it at all. I've just moved my face. I don't know, I'm a little bit more than a foot, maybe 20 inches or so, and then I just move further away, and bam, all of a sudden the O just pops out, and the optic nerve is creating this in the retina, and the brain just fills in the blank with whatever the background is. I used to have a figure in the classroom, it disappeared though, that showed like really strongly patterned background, and your brain would just fill in the pattern right there where the blind spot is. But anyway, it's kind of curious. You should be able to see it by moving your face forward and backward, the distance between your nose and the screen, until you get it just right. And so those are just a couple of examples of non-ideal design in the spirit of Stephen Jay Gould. And biological evolution is a fact. It has been factually observed, factually established that the genetic structure of populations change from generation to generation. That is biological evolution. It occurs. There is no denying that it does. Now, the mechanisms by which gene frequencies change can be hotly contested at times, even by biologists that disagree with one another. Charles Darwin proposed one mechanism of change as natural selection, and we've described this. Other mechanisms of evolution are also known to occur. Remember, evolution in this case means change in the genetic structure of a population over time. So this next slide is kind of an important one, because it shows you 5 forces that most biologists would readily agree are capable of causing evolutionary change. They're not in any particular order, although we have put natural selection as number 1. This is what we've established. But we can also have sexual selection or mate selection as number 2. We've talked about that already. We haven't really talked about gene flow that much, but gene flow is simply immigration in or immigration out of a population. So individuals entering or exiting a population can change the genetic structure of that population. Mutation, certainly if we have a high mutation rate, then we can have new genetic combinations which are arising, and that could cause a change in the genetic structure of a population. And the last one is genetic drift. And so all 5 of these are capable of producing changes in the genetic structure of a population. And so we have discussed the majority of these. We did discuss how mutations arise, and we did say that they are the source of variation, genetic variation that evolution acts on. But we're going to right now look at genetic drift, and we'll define that one. That's one we haven't touched on previously at all. Most of the others we have before. Number 3 we really didn't, but it's kind of common sense. You see it and you understand it. Okay, so let's look at the role of chance in evolution. Sometimes people that are not knowledgeable about evolution will try to dismiss it as purely an idea of random chance. And this is pretty much the opposite of what natural selection is. Natural selection is an organizing force. It leads to adaptation. It leads to a fit between a lineage of organisms and an environment. That's not random. That's a directed process. But chance does have a role in the process of evolution. And what my intention is to point out to you exactly what that is. So first, chance has a role in mutations. Mutations occur randomly. They don't occur because of need. If it's getting colder, you don't get more cold mutations for cold adaptiveness, or the opposite way either. Natural selection simply acts upon the mutations that are there, and the mutations that are there occur randomly. And so there is an element of chance there. A second manifestation of chance in evolution is in the concept known as genetic drift. And I'm going to define genetic drift for you on the next slide, so hold your breath. But genetic drift is a random force. It is an agent of evolutionary change. We saw it on that previous list. And genetic drift is much more likely to impact small populations. And besides these 2 factors, there's relatively little randomness or chance in evolution. And so it's kind of interesting. So let's go ahead and look at genetic drift. This is a definition. It's random fluctuations in allele or gene frequencies over time due to chance. So fluctuations in gene frequencies over time is simply evolution. So it's evolution due to chance is all that genetic drift is. And this can occur in many different ways. As I said on the previous slide, and I repeat here, small populations are likely to have the frequency of alleles in their gene pool affected by chance. And you can see that this example down here with the flowers tries to illustrate that with just a few reproducing. It actually gives you allele frequencies. So that, remember, allele frequencies have to add up to 1. And so this is showing the frequency of the R, the big R, is 70%, and 0.3 is 30% frequency for the small r allele. It's not the red and white flowers, it's the alleles, the big R's and the little r's. And if 5 of 10 plants leave offspring, and it's kind of random as to which ones have offspring, we could have this second scenario here. So this is time 1, time 2, time 3, and there are 2 reproductive events. And again, you can, just by which plants leave offspring in particular years, you can completely lose an allele. And that's what happens if you notice at time 3 here, that 100% of the alleles are big R alleles. And that's just because this is a small population. Note that there's only 10 individuals in this population. Now, if there were 10,000 individuals, this wouldn't happen. You might have a change and lose, a few alleles, but because the population is so large, it wouldn't be impacted. And so that is exactly why small populations are more impacted. You can also consider the example of human birth ratio at 2 hospitals. A hospital in a large city where there may be 300 births in one night, or a hospital in a small town where there's, say, only 4 births in one night. And in the big city, you'll probably have pretty close to 150 male babies and 150 female babies. But with only 4 births occurring in the small town hospital, you could have 3 females and 1 male, or 3 males and 1 female. That's 75% of one gender and 25% of the other. And you can see that's a departure, a statistical departure from the expected 50% probability. And you get these departures in small populations because the sample size is so small. And genetic drift can result in the loss of alleles in especially small isolated populations. So let's look at 2 more forms of genetic drift. The definition of genetic drift doesn't change, but population bottleneck is a specialized form of genetic drift, as is founder effect. Population bottleneck is when environmental factors cause a severe reduction in population size and in genetic variation. This happens at the same time. And later the population may increase in size, but it's easier for a population to grow than for genetic variation to accumulate. It takes a long time because it starts with mutations, which have a relatively low rate of occurrence. Here's an example that will visually bring this home to you. This is a population bottleneck. So it shows the original parent population, and the different colored marbles represent genetic variation. And so we can greatly reduce the population size, just put a few here. And even though the population grows later, notice that we lost the red allele going through the bottleneck and doesn't restore, even though the population grows larger. And so we can see time 1 pre-bottleneck, time 2 during the bottleneck, time 3 post-bottleneck, and you get the idea. Here's another way of displaying that in a graph, where the width of the graph shows the size of the population. And we can see before the population bottleneck, there was large genetic diversity. You go through the bottleneck, and even though the population regains a size similar to what it was before, the genetic diversity remains low. An example of this was the northern elephant seal, which occurs on the Pacific coast of North America. It was almost hunted to extinction in the 1800s. The population was reduced to only around 20 individuals on one island off the coast of Baja, California. The seal population, elephant seal population, has subsequently increased to tens of thousands of individuals. But because of this bottleneck, they don't have very much genetic variation. What is interesting, this shows 2 males in combat, and they really go at it. They'll be bloody at times. They set up a territory on the breeding beach, and if another male enters it, they go charging over there. It's so much so that I've actually seen one of the offspring of the male be killed by that male as he just is oblivious and runs over the baby seal as he goes to attack a male that goes into his territory. So these are really quite imposing animals. The males are huge. They have these funky noses. They don't really look like elephant trunks, but that's why they were called elephant seals, because they have these funky proboscis, and really interesting. A second example of a population bottleneck was the population of cheetahs. And cheetahs, of course, I've used as an example of a very fast animal, the fastest land animal, running at 60 miles an hour. The natural cheetah population underwent a genetic bottleneck 10,000 years ago. This was not because of humans. We're not sure what the cause was. It's kind of interesting, though. And this, again, is another way of graphing it. So where we have 2 lines, the blue one for population size and the red one for genetic variation, and it shows over time that they went through this bottleneck event, and both variation and population size dropped, but population size climbed back up, but variation stayed low. It takes a long time for genetic variation, for mutations to accumulate and get genetic recombination. So now cheetahs are severely inbred, and they have very little genetic variation, and the males often produce malformed sperm. Here's a 2-headed cheetah sperm right here. Because it has 2 heads, it can't swim properly, and it can't go up the female reproductive tract to fuse with the egg and fertilize it. So cheetahs are in real trouble in the modern world. Another example of the lack of variation in cheetahs was at a breeding zoo where there were all sorts of different big cats, and a virus swept through the cheetah population, and about half of the cheetahs were killed. But of the lions that were in this same breeding facility that have much higher genetic variation, none of the lions even got sick. And so this can show you the value of genetic variation. A second manifestation of genetic drift, and this is in fact a subtype of population bottleneck, is called founder effect. And when we say founder, this is like founding a new population. So founder effect is when a few organisms with a limited subset of the original gene pool establish or found a new isolated population. And the result may be higher than expected incidence of a particular genetic condition that was present in one of the founders, or maybe several of the founders. And this is especially true if the population continues inbreeding, doesn't have new genes come in from outside. And so this actually shows the original population made of blue and red dots, and we take random subsamples of 5. We could get 5 red ones, we could get 5 blue ones, or we could get a mix. And the mix would best represent the population. But when you get a founding event, you don't choose. You just get some organisms, and they may or may not mirror the original variation or the variation of the original population. An example of this among humans is the Amish population in the United States. This population was founded by a relatively small group of individuals, and within those founding individuals, there was at least one person that had the trait of polydactylism, having a 6th finger, and this is actually a dominant trait. And so polydactylism, we can see, poly means many and dactyl means finger. And we look at the x-ray down here, 1, 2, 3, 4, 5, 6 fingers. And so among the Amish, there's a higher incidence than in the general population of polydactylism. Founder effect is a special case of genetic bottleneck because the bottleneck event is the founding event. If you think about that, you'll understand it. Okay, finally we get to patterns of evolution, which was the name I gave to this module. And in patterns of evolution, we can see several possibilities. One is non-branching evolution, in which one species changes to another over time. You could draw or write the letter A, make a single arrow to the arrow B, to the letter B, and that would show that species A turned into species B over time. Kind of here. This says, I think, straight line or something like that in Korean. I don't read Korean at all, but I had a Korean student tell me that. A second possibility for a pattern of evolution is you can have one lineage, lineage A, that gives rise to 2 lineages. It splits, it divides. And so even though we might have A and A continues, and we also get B branching off. And so this might say branching or something like that in Korean. And the original species doesn't have to survive. It could change. A could go to B, and then also C could branch off, but A could survive, and then B could also branch off. So this shows 2 patterns, possible patterns of evolution. And so by having multiple branching events, these are speciation events. We'll talk about how speciation might occur in the module after this one. A third pattern of evolution is called adaptive radiation. And this is when a single lineage, as shown in this picture, one ancestral species, gives rise to several or different lineages. And so that one lineage diversifies into many. And so if you're doing this with letters, you could say species A gives rise to species B, C, D, E, F, G, H. And it doesn't the dividing events, the speciation events, don't have to occur all simultaneously. They can occur at different times, as you can see here. There are 2 common causes that you should associate with the pattern of adaptive radiation. The first is called ecological release, and that's if that original ancestral species is somehow isolated. Perhaps it goes to an island or somewhere, and there it escapes predators, competitors, pathogens, other ecological interactions are reduced in their intensity, and it allows that single lineage to become multiple lineages. So that's one causative mechanism that could give rise to this pattern of adaptive radiation. A second one is called evolutionary innovation, Well, innovation means coming up with something new. So evolution comes up with something new. Think, for example, back to when insects evolved the ability to fly. Before that, they couldn't really fly. They might be blown around a little bit by the wind, but when they developed wings and they started to really capitalize upon this ability, then all of a sudden insects were mobile, and they actually underwent a huge adaptive radiation and also a coevolution with flowering plants. And now that insects could serve as winged pollinators and move between plants, there was a lot of evolution. Another structure here that I listed here is the radula of mollusks. In the ancient seas, the animals without backbones, the invertebrates, none of them had hard mouth parts. And then in that context, one lineage of mollusks, they came up with a hard mouth part called a radula. It's like a biological file. It's made out of chitin, that complex carbohydrate. It's a structural molecule. It can be used to rasp algae off. It became a drill in some, could be used to drill through the shells of other invertebrates, and it even became like a beak-like structure in some of the octopuses and other cephalopods. And so it allowed the radiation of mollusks, which is one of the more diverse non-insect invertebrate groups. So again, it did give rise to them. Another example of adaptive radiation is the fruit flies, or the Drosophila, on the island of Hawaii. There's something like 500 species that have colonized back and forth among these islands, and they change to one species and they go back to the island where they originally came from. It's really quite a complicated system, but nevertheless it shows that. And it can be things like the pitch at which these animals kind of buzz or have the whine. They go bzz, bzz, different pitches, and it makes a difference. Or different dances that they do, different coloration, spots on the wings, different colored eyes. All these things can lead to speciation events. And so because there weren't a lot of insects on Hawaii, their ancestors encountered many empty habitats. And now some of them are predators, some are parasites, some are herbivores, so they've filled a variety of different niches as well. Also on the island of Hawaii, a classical example of adaptive radiation, and this would be ecological release, as with the previous example with the Hawaiian fruit flies or the Hawaiian Drosophila. One finch species is believed to have reached the Hawaiian Islands, and they underwent these wild modification of the beaks, and these beaks are long like this, some of them, because they're nectar feeding birds. Finches were originally seed eaters, but here there were no nectar feeding birds, and so it was an empty niche, and so some of the finches evolved to fit that way. And so there's many lineages here. It's interesting that in addition to these that are shown here, over 20 different honey creeper species have become extinct. So we're talking, of course, this occurred over millions of years, but nevertheless, it is interesting. And so here's Charles Darwin. He's doing tree thinking. There he is. This is actually notes from Charles Darwin's, one of his notebooks. And he says here, I think. And he shows this pattern of species, and there's his finches shown over his head there. So kind of interesting. So it shows an ancestral species down there, and then it shows some of the descendant species out there. And just like the finches, one species comes in, and there are, in fact, I believe 13 species of Darwin's finches. A very famous book called The Beak of the Finch by a modern author describes this group quite well. It's a very worthwhile book to read. If you're at all interested in evolution, you'll find it fascinating. Again, Darwin's finches, are a classical example of ecological release. Not as diverse as the honey creepers, but they haven't been on the Galapagos as long as the finches have been on the Hawaiian Islands. Here, we believe it was a South American ground finch which colonized here and then diversified into these different species. And you might be going, well, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Well, in fact, those are the 13 that are found on the Galapagos, and it is believed that a flock of warbler finches colonized another island group called the Cocos Islands, where they have become separate species. But this is just showing the Darwin's finches. And so it's kind of interesting to consider this. This shows all the finches. It shows they're divided into different groups: seed eaters, flower eaters, and insect eaters. And so there's our South American ground finch, and we can see that they've is given rise to all these other forms. And here's a quote from some of Darwin's writings on the finches. He says, "Seeing this gradation and diversity of structure in one small, intimately related group of birds, one might really fancy that from an original paucity of birds in this archipelago, one species has been taken and modified for different ends." Indeed, we see some interesting things. If we look here, the woodpecker finch actually uses a tool. Because real woodpeckers have modified tongues that's really long and have barbs on the end, so they can reach in and get grubs out of their galleries in the wood of dead trees and under the bark. But this finch has no such tongue, so it uses a tool. It's kind of interesting. The sharp-beaked ground finch, Geospiza difficilis, is also sometimes called the vampire finch because it will at times feed on ectoparasites on some of the seabirds that come in, like ticks and other things. And of course, it gets a tick full of blood, and every now and then it will miss the tick when it's pecking, and it pecks the bird, and it just directly will drink the blood. Of course, on an island, water and protein are often in short supply, and so drinking blood is a solution to that. Anyway, this is a very interesting group of birds. Darwin didn't realize their importance at the time he was on the Galapagos, and he had to go back after the fact because he neglected something important, which is really unusual for Darwin. Just shows he's human like the rest of us, I guess. But he neglected to label which island he got which birds from, and he had to go back and do that later once he realized their great importance. Anyway, we can see those. Okay. Finally, before we wrap up this module, rates of evolution. How quickly does evolution take place? There are two extreme views. Darwin believed that evolution primarily occurred gradually, and so this is called gradualism, constant gradual changes in species over time. You can see that evolution or change over time, and we can see a nice straight line. But some of the paleontologists, in studying different groups of fossils, found that they didn't agree with this. They found another idea. They saw that when they looked at some groups, that evolution seemed to occur in spurts, and there were periods of stasis, which are these horizontal lines, and then the little spurt is in between. And so they call this punctuated equilibrium. Most of the time, the species were in stasis, were relatively unchanging. Stabilizing selection was in action, and then there were periods of change in between. So these periods of stasis were punctuated with periods of change, and the change, of course, occurred. There may have been environmental change, and then there was very rapid directional selection. Then they leveled off again. And so you'll notice that you can get as far using either technique. You can get up here by going with gradualism, or you can get up here by going with punctuated equilibrium. And so it's not a question of which one occurs, because they both occur in different lineages. And this is just showing it with this kind of cartoon picture with birds. Horse evolution shows some gradualism. Ancestral horses were tiny forest dwellers with 5 digits, and their body size gradually increased over time, and their number of toes decreased over time, and their legs lengthened over time. And now the modern horse runs on only its middle fingers. In trilobite evolution, we can see punctuated equilibrium as species appear abruptly in the fossil record and then remain unchanged for a while, and then more species appear. So examples of both patterns. And with that, we're going to conclude this module.